I'll be starting in next 5 minutes. So let's wait for 5 minutes so that people will join. I think let's start. So, good evening, everyone. I'm Shikha Kaushik. I'm your teaching assistant, and we are studying structural biology subject. So far, I have taken you through all the structural biology techniques where we talked about X-ray crystallography, NMR, cryo-EM, spectroscopy, and microscopy. And in the last class, we started about various molecular visualization softwares where we talked about different why there's a need to do molecular visualization. Why do we want to see? Why do we want to understand the structure of these macromolecules? And what is the need uh, so as to understand biology in depth. So basically, molecular visualization is helping us to understand the structure of these molecules, and from the structure we are able to understand what are the function associated or the different region which are leading to the activity of that particular protein or macromolecule. So I have asked you to go through the molecular visualization software. I have told everyone that there are certain commands that you can use to make your structure look much more better depending upon your requirement. So I have given you so many commands and things. Also, sir has mentioned in his PPTs where he has explained you how you can do or how you can change certain thing in your structure using PyMol. I'm right now focusing on PyMol because that is what we use maximum in our day-to-day molecular visualization studies. But yes, we can work on COOT, we can work on RASMOL. There are other molecular visualization software that are also being used in uh, molecular dynamics study. So coming to the so I'm not taking you, I'm hoping that you guys have checked all the visualization software, especially PyMol, and worked on various commands so that you can understand how you can visualize your structure in a better form, or how you can understand what all residues are involved in interactions, or what are the hydrophobicity or polar site, or what all type of amino acids are present in your structure, and what all regions you can code, where you can or you may want to understand the active site, and you want to understand where the active site could be. So there are so many things that you can do through these visualization software, and they are very important when we are talking about molecular dynamic or molecular simulations, or molecular docking. And in today's class, we will be studying molecular simulations. And as the name suggests, simulation is something where you are mimicking a system. And molecular dynamic simulation can be defined as a process to study and analyze the dynamic nature of system, where the system can be small as atom and to as large as a galaxy. So basically what we are doing a molecular, if I break this term, molecular simulation. Molecular means something at a molecular level, something at a smallest level of that particular system, and simulation is basically mimicking that, but mimicking any of the system. So molecular simulation is done by using computer simulation, and with the help of computer simulation we try to understand any of the system that we are studying. So if I talk about computer simulation, it is a process of mathematical modeling performed on a computer, and which is actually designed to predict the behavior and the outcome of a real or a physical system. So what exactly we are doing that, so if you imagine this thing like this, that there are so many things that are happening around us, or inside our body. So we have things that are happening, which are natural processes happening in a real situation, real condition, and we want to study those systems. We want to study those processes. So molecular simulation, through molecular simulation or molecular dynamic simulation, we are able to study those systems that are happening in nature, or in a real scenario, or inside the organism body on computer. And that is what is something revolution in terms of scientific or technological achievement that we have achieved as a human so far. So molecular simulation is something that has made it easier for us to understand any system, to visualize the things on a computer, on a mathematical program, uh, where we can understand by changing, altering certain condition, and we can simulate the real system as it is. So definitely it's not going to be exactly the real system, because there are many other forces, external forces. There are many there are huge forces involved, but something to to make it visual on the computer screen, on a screen, is very fascinating as a researcher to me, and I think to the entire world. So if you could look on this cartoon, and if you could see it, so this actually depict a simulation, molecular simulation of nanometer system, and basically it is showing the kinesin motor domain docking on a tubulin heterodimer. So can you imagine something that is happening at a very small level, at a very small scale, because we have cells of range of micrometer or nanometer, and something that is happening at the scale of nanometer can be visualized on a computer screen, and we can understand what all things are happening at that particular place. So this is just a depiction, this is just a cartoon showing what all we can visualize using molecular dynamic simulation. So if I talk about a journey for a real system to a model system, and that is happening through computer simulation. So we have a real system. So we have a real system, and we have to make a model so that I can generate a model system which is exactly like a real system. So real system, they are thing when we work. For example, if I give you an example of a laboratory, we perform certain experiments, right? And we try to mimic a real system in forms of experiment, maybe through in cells. Cells, when we are working on cells, that is also not a real system. We are using cells as a model, performing experiment on that cell with this thing in our mind, that these cells are actually depicting cells that are present in a human body. So I'm just giving you an example. So real system is something where you can perform uh, where you perform certain experiment, you get certain experiment result, and you have something to talk about for in reality for that particular system. But when we mimic this model, when we make a model to mimic this real system, we generate a model, we perform certain simulation, we have certain we also construct certain theoretical speculations. We have certain theoretical speculation for that. And if we do certain simulation, we have simulation result. Or if we are using some theories that are done through some hypothesis, theoretical laws, and we are trying to prove some hypothesis, that is that leads to theoretical prediction. And then once we have all the thing at place, we compare our generated model with the real system, and with the help of theories, we compare this generated model with the real system as well as with theory, and try to improve our model. And we try a level best to take this model to towards reality. So as much as we can simulate a reality that makes a model better. So I'm not saying that uh, so far we have achieved exact real models, but we are very close to that. So whenever we do something through molecular simulation, we try to validate that through a real system as well. So that is something we do, but definitely molecular simulation is helping us to have so many things solved through computer, that we don't have to actually generate a real system to perform experiments, and to because whenever we perform certain experiments, or whenever we are trying to understand real system as it is, there are so many forces that may lead to the failure of that particular experiments. But when we are doing something with the mathematical modeling, with the mathematical coding or softwares, that is not, because machine is something which is programmed to do certain action, and the chances of error are there, but we can try to minimize that because there are no human error involved in a machine. And I'm not saying one system is better than another system, because both the system right now is important to understand what all we can achieve to through them we can achieve a lot of things to understand biological system. So I'm not saying that one is better than another, but definitely both the system are important to understand actual processes that are happening around or within us. So if I talk about you know principal tools in theoretical study of biological molecules, so molecular simulation is one of that, and it actually helps us calculate the time dependent of any system. So it not just giving you the actual not just showing you the actual scenario, but also through molecular dynamic simulation you can change it, make certain changes. You can fluctuate your parameters and try to understand what all possible changes happen in a real system, and it no doubt it helps you understand so many things. So if I talk about some experiment which are impossible to study. For example, it is impossible to study what is happening inside the stars, or sometimes it is impossible to even do the forecast. So that might not be done through molecular simulation. Some experiments are too dangerous. For example, flight simulation or explosion simulation, some of them are very expensive. When we talk about high pressure simulation or wind channel simulation, they lead to a very expensive, they require very expensive system. And there are some blind experiments as well, where you cannot observe very short time scales or very small space scales parameters. So these are the things. There are certain limitations, but definitely molecular dynamic simulation is something we really want to understand. We really want to use to understand the system that are and processes that are present or happening around us. So if I divide, why do we need to simulate all this, and especially when we are talking about biology? So this actually replaces the experiment. As I mentioned, you don't have to perform the experiment, because while performing the experiment, you might have some human error that leads to that will not exactly represent the actual system. So if you make some human error, you will have some different kind of result. But when you do molecular dynamic simulation, you don't have to perform experiment. You don't have to do something that may lead to lot of money, lot of reagent, and can can have a failed result due to some human error. So it actually replacing the experiment, and you can do these experiment on your screen. It provoke experiment. It definitely make you more curious. It actually give you so many information that you can think of what all can be done to understand this particular system. And definitely it explain experiment. It also give you all the I hope my screen is visible, right? I think there's some kind of network issues. Just give me a second. Uh, is it connected? Yeah. Okay, I'm so sorry for this thing. Yeah. So yeah. So it also help you to explain experiments. So it will give you the details, what all can be done, what all can be achieved, because it is actually making the exact scenario. So you can perform your experiment, or you can understand your experiment on a computer screen by changing the parameters, by changing the conditions, by changing the conformations. You can understand what you are going to understand through an actual experiment. So it is more realistic simulation method. Molecular dynamics give you a more realistic simulation method. Whatever we perform in lab, whenever we do experiments in lab, that is also kind of a simulation, because we are not working on exact human body. We are trying to mimic the human body in term of our cell cultures, in term of our expression systems, and so many other things that we do in a laboratory. But this seems to be a more realistic. This is one of the more realistic. This is not the most realistic, but it is one of the realistic simulation method. It can provide you structural and dynamic information. It can also help you understand the native condition and the non-native conditions as well. So not just the native conformation of protein is something we are studying. We can do certain changes, and we can understand what all possible conformation this protein can achieve if there are some kind of changes in the amino acid sequence. So the non-native conditions are basically the condition which are not present in reality. It can also give you time-dependent behavior of a molecular system. You can study structures, dynamics and thermodynamics of biomacromolecule complexes, conformation changes of proteins, or nucleic acids and whatnot. You can actually visualize these things on your computer screen and make some changes to understand all these big biomolecule structures. So this is one of the very important tool that we as a human, or we as a researcher, are able to make. Obviously the credit goes to all the people who have worked in this particular field and were able to make these tool for us. to study. So I think it is, if I look back, and if I have to go back in a time where these tools were not there, I can't even imagine life as a researcher to be, because it's very easy nowadays to understand structure, function, or entire protein structure and its conformation, so easily available on internet, and you can use that information to perform your experiments. So if I talk about the application of molecular dynamic simulation in biology, it helps you to understand protein stability. It helps you to understand what all conformation changes can be possible. It helps you understand protein folding, molecular recognition, proteins, DNA, membranes, complexes. It helps you understand all these structure in details. So you can visualize all these things. You can understand this system. You can actually understand what happened when a ligand interact with the receptor, when a membrane, when a cell come in contact with any of the virus or any of the pathogen. All these things can be visualized with the help of molecular dynamic simulation. It also tell you ion transport biological system, drug designing can be achieved, and you have X-ray and NMR crystal structures that help you understand how you can design a drug against that particular protein. So if you talk about COVID-19, when there is a COVID-19 and when we don't know what this virus is all about, with the help of molecular dynamic simulation, we can achieve possible drug targets or repurposing of certain drugs that can be used to fight against COVID. So with the help of these tools, it is really helpful to understand the structure or the biological process or the therapeutic aspect of understanding these structures. So no doubt molecular dynamic simulation has created a lot of, it has opened a lot of doors for us. So if I talk about a bit of history of molecular dynamic simulation, especially in the field of biology. So the first protein simulation by McCammon appeared in 1977 with the simulation of bovine pancreatic trypsin inhibitor. So trypsin is a protein that cleave your trypsin is an enzyme that cleave your proteins that actually cut the lysine, it actually cleave your protein and the lysine residue. So the first simulation that has achieved by McCammon in 1977 is an inhibitor that is blocking a pancreatic trypsin. And then there are people in 2013 who received Nobel Prize in Chemistry, Karpus, Levitt, and Vassell. They actually discovered this tool which is called Chemistry at Harvard Macromolecule Mechanics, which help you to study, molecular simulation, and that is CHARM, which is called CHARM actually, and they received Nobel Prize for this discovery of theirs. So if you look at the, visualize, this is just a cartoon showing how we have achieved from 100, we have actually able to visualize structures at a very small size. So initially in 1990s we could able to decode the structure of lysosome only, but now we have reached to a stage where we have so many structures. And if I talk about just PDB, we have 1,70,000 around structure, out of which 30 to 40,000 are unique structures. So we have came so far with the help of these software, structural biology techniques, and these molecular simulations, tools. So if I talk about the problems in biology, so whenever we talk about uh, something that is new, something that is providing us, that is solving our problem. So we need to address we actually, with the help of these tools, we address certain problem. And there are so many problems in biology, especially when we are studying the system at a laboratory level. So Protein folding is one of the very faced problem whenever we are performing our experiments. So with the help of molecular simulation, with the help of molecular dynamic simulation, we are able to achieve, or we are able to understand how these protein folding happen in our system. So this tool has made us understand exact process of protein folding. So that is something very important to understand. So it is very difficult for you to understand how the protein is getting folded in your laboratory. You cannot visualize it, but now with the help of molecular simulation, with the help of your computer, you can understand at what particular residue the protein is bending, what all residues are present in helix, what all beta sheets are there, what kind of conformation this protein is achieving, what is the active site of that particular protein. All these things can be done with the help of molecular simulation. So that powerful this tool, that's how it has created a lot of revolution in the field of science. Then we talk about protein ligand interaction. That is something we really want to understand. We know that there is a ligand, there is a receptor. So that receptor, that ligand bind to a receptor, and there is some function that happen. There are some protein that get activated. There are some downstream protein that is affected by the interaction of this ligand and receptor. So that these all interaction now we can visualize with the help of molecular dynamics. So we can understand what is the active site, what are the whether there is a pocket involved, whether there is some kind of a region where this ligand is binding, and what all things can be done if we have to block this interaction. These all things can be done with the help of molecular dynamics. So if I talk about the comparison in vivo, in vitro, and in silico, so we have in vivo. In vivo is what our system is. So whenever we do something, whenever we, for example, if I express a protein in an in vivo condition inside a cell, that can be called in vivo. There are thousands of proteins that are present. There are not even thousand, but I think in millions of proteins that are present in which that particular protein is getting expressed. So we generally cannot study in vivo. We have n number of protein out there, but in case of in vitro we do cloning. We put a gene of interest in that particular expression vector. We try to express this particular gene or a protein in large number of amount, and we get some, we purify these protein, and we have the purified protein. So now we have a controlled number of proteins we have control. But in vivo we don't have a control on the number of protein, we don't have a control on type of protein. There are millions of proteins that are present inside the cell, and we cannot regulate that. But in vitro condition we still can actually try to make one kind of a protein in large number. But in case of in silico, we just study one or two molecule of that particular protein. So we have narrowed down from large number of protein to one with the help of this in silico study, or you can say molecular simulation. So now initially we have heterogeneous mixture of protein where we cannot control other proteins that are having an effect of effect on a particular protein. But in case of in vitro we have homogeneous protein structure where we have a lot of number of a same protein, but in silico we can study single protein molecule. So that's how that far we have came, and we can now understand a single molecule. So if I talk about the time frame in MD simulation, generally we collect the frame after 1 femtosecond, that is 10 raised to power I think 50. So can you imagine that you can understand the changes happening at that particular level, at that particular time frame. So typical and length scale of a different simulation techniques are given in this particular cartoon, where you have quantum mechanics, where you can achieve beyond femtoseconds. Then you have molecular dynamics, or yeah, molecular dynamics and other system that you can achieve at the femto or picoseconds, and then you have your, some limited the different type of molecular simulation techniques that help you achieve something at a nanoscale and microscale as well. So this is how this actually helped us to understand a system at such a small scale, at such a small time. We can decode the changes or that is happening at a very small, I mean, imagine that is happening at a femtosecond. So that is something is commendable. So this is what molecular dynamic simulation can provide you. So if I talk about the force field is something we say is a collection of equation or associated constant designed to reproduce the molecular geometry and selective property of a textured structure. So we have certain force that is present in whenever a system is working. There are some force and that leads to changes in that system, and that is how process work, and that is what we want to understand. So for example, if I provide a force, there can be bond stretching, bond bending, there are some torsion angles that are being changed, there are van der Waal forces that are present in a particular system. And when I'm talking about system, please imagine it like a biological system or something happening at the molecular or atomic level. So these are the forces that are present in that particular system, and this is how, that is what we try to understand, because when we talk about the molecular simulation, they follow the Newtonian mechanics. So force is always there. There is some force, that is why there is something happening in a system. If there is no changes, no force applied, or nothing is happening, then the system is then we don't have anything to study, to understand. So these are the things that are there. These are the parameter that we select. These are the property that we study, and that's how we test our system. So these are some forces field. If I talk about the bonded interaction where two bonds are involved, so we have bond, we have interaction, and because of this bond, there are some changes, there are some movement, there are some energy associated with it. So that is what happened when we talk about bonded interaction, where we have energy bond is equals to kbr minus r naught. R minus r naught is the initial distance, or you can say the lowest distance, and r is what we achieve at the final stage. So these are the changes, these are the movement that is happening at the molecular level. Then we have angles associated, where we have some dihedral angles as well that I'm going to talk about. So there are so some bonds are formed in which the angle, there are some linear bond, there are some bending form bonds. So the bonds that has some angles, their energy is given in the form of angular, the angle they are making. So when we have linear bonds, that is given by a distance. When we have angular aspect, that is given by angles involved. And when we talk about dihedral angle, then we have our psi and phi, which we talk in when we are studying proteins. So in case of dihedral angle, we have psi and phi. Phi is angle between C and N, and psi is angle between C alpha and C main C. That is that is this one. So these are the angle that we study, that is very important. These are the parameter that is very important when we are simulating any of the system. And if I talk about the non-bonding interaction, there are some non-bonded interaction like van der Waal forces interaction, Coulombic interaction, hydrogen bonding, and they have their own parameter that we use to understand the energy changes or movement changes. So I'm not getting into detail of these things, but if you could see, they are very simple to understand. So these are some molecular representations. Oh, sorry, these are actually the mathematical representation that is telling about the changes that is happening at the energy or the momentum level. So what are the major steps of molecular dynamic simulation? So we have certain steps that we follow to achieve molecular dynamic simulation. Whenever we are simulating any process, any system, we have to first build a realistic atomistic model of the system. So first, so this is like something when you are making a drawing, when you are having a sketch, you have to first understand what are you making. So if you are trying to make a portrait of a person, you have to understand what exactly the object or your subject look like. So you have to, likewise, when you are simulating a system, you have to actually understand the realistic aspect of that particular system. Then only you can make a model of that particular system. So first, build the realistic atomist model of that system, then simulate the behavior of your system over time using specific conditions like temperature, volume, pressure, etc., etc. So there are so many parameters that actually have an effect, change, or anything that is leading to a behavioral that is changing your system is what you try to simulate. So first you make a realistic system, then you change the parameter that are present in that particular system like temperature, pressure, and volume. Then you analyze the result obtained from molecular dynamics and relate it to the macroscopic level properties. So then you try to, actually you have realistic system, and then you try to improve your system, or you try to analyze your system, that whether it is mimicking with the macroscopic level of that particular realist from your real system. So you compare that thing. So these are the major steps that are there in your molecular dynamics simulation. So this is just this is just a representation showing how you do molecular simulation, especially when you're studying a protein, and that's how you have your protein structure from your protein data bank structure. Then you change it, then you make a changes, because to understand a system, for example, if you are trying to understand what all changes lead to the inactivity of a particular protein, then you have your you have your original structure, original protein conformation. You insert certain mutation with the help of computer, with the help of molecular dynamic tools, and then you try to understand what all structural changes that a particular protein has achieved due to these mutation, and then you try to align both the structure and understand that what all changes leads to inactivity of that particular enzyme. So this is just an overview to understand what all things you have to do when you are doing a molecular dynamic simulation, especially in terms of proteins. So how do you build a realistic atomist model of a system? So first thing is we need to determine a valid protein structure. For example, like I gave you an example of an enzyme, inactivity of enzyme due to mutation, for example. So before understanding the inactivity which happened due to a mutation, you need to have the original structure, and that original structure you can achieve experimentally or you can achieve theoretically. Experimentally, that you have available databases where you have X-ray crystallography structure achieved from X-ray crystallography, NMR or cryo-EM, and that file you can extract from the database available, which is Protein Data Bank, and you can download the file. That structure is actually validated or achieved experimentally. But when you don't have any structure, when you don't have any protein structure available, then you try to achieve a structure of that particular protein through theoretical methods. And you have three types of theoretical method, where you have homology modeling, fold recognition, and ab initio modeling. So in homology modeling, you try to look for homologous structures with the help of, you search your protein, you have your protein sequence, and you try to look for homologous structure or homologous protein that are present, and with the help of that particular protein of a different organism, you try to model your protein of interest. And fold recognition is something that is not, I mean, people are still working on it, and I don't know whether I think it is not that much used, but ab initio method is something that is specially done in terms of peptides whenever you're doing some drug designing, because they are small, or they are something that does not have any similarity with the existing proteins, and that's how you start building a structure of that particular protein from the scratch. So you might have heard about Rosetta and ITESR. These are the software that we use, and nowadays these software use all the three techniques to achieve the final structure. Because this is not something. Sometimes you don't need to. Sometimes you have structure. You have some homologous aspect of that particular sequence, so you can achieve a homologous. You can achieve a model of that. You can achieve a structure through homology modeling, but sometimes you need to apply all the three methods to have your final structure. And we have softwares, we have methods, we have system generated which help you and use all the three techniques simultaneously. Once we have the structure, we need to perform structure repairing. So what is structure repairing? We have to, whenever you open your PDB file, that I also mentioned in your in the last class, that there are some region that you need to look. You have to understand whether all the residues are present or not. If they are not present, you have to identify the missing residue, missing atoms, and you have to put those those missing residue there. Then you correcting wrongly modeled residue group, because there are possibility that these files does not have the correct model, correct alignment of residue. So you have to do all the correction. You have to correct the steric clashes. Then we need to clean the structure. So we get the structure, and we have to delete the hydrogen bond. So many of the PDB file does not have hydrogen bond, because most of the protein structure are achieved through X-ray crystallography, and X-ray crystallography does not provide you a structure of hydrogen, water molecules in the system. So, but there are some PDB file where you have hydrogen molecule, or you have solvent present like water. So you have to delete all these things to make it a clean PDB file. So you have to delete hydrogen bonding, you have to delete solvent, you have to delete other unwanted factors that are there because of maybe because of the crystal you have made, maybe because whenever you are doing your NMR, whatever whatever happened due to the solvent presence, these things you need to remove from your PDB. You have to make your file clean, and that's how you clean your system. Then you have to simulate the behavior of your system over time using specific condition. So now you have your clean PDB file. Now you have to prepare, because this PDB file will not give you a simulation. You have to change your PDB file to a topology or parametric file. You have to make it a topological and parametric file, and for that you have to do you have to follow certain steps, and that's how you achieve. And whenever we talk about molecular simulation, it is basically based on Newton's law of motion. So you have force applied to a system, and that force lead to certain changes, because there for every action there is some reaction, and that's how that is what is a basis of molecular simulation. So you have to change all the you just give me a second. I think there's someone at the door. Please just give me a second. uh, Wait a minute. Okay, yeah. So yeah, where were we? Huh. So as I mentioned that we use Newton's second law to calculate the acceleration on each atom. So we use these. I mean, I'm not getting into detail of this, but I think Sir has talked about that we use Newton's law and we try to iterate and iterate and iterate and integrate these laws of motion so that we can understand what all changes can happen because we are simulating. So we are providing a force. We are providing a change for something that is changing our particular system. So whenever you have something that leads to change, for example, you are providing a force, there will be a reaction, and that is what you want to understand. That is what you say when you talk about studying a system, because you want to understand all the parameters, all the things that is happening in that particular system that leads to either some function or either some changes. So what we require now we require atomic connectivity information to tell the MD program that one atom is bonded to another. We need to understand the bonds present. We need to understand what are the possibility that leads to breaking of this bond or making of this bond, and that is done with the help of changing that we have to do, that we have to achieve, and we can achieve that once we change our clean PDB file to a topology or parametric file. So, uh, and next thing is the spring stiffness and the equilibrium of the bond length. So, okay, fine. So, if I talk about for example, if I'm talking something at the atomic level, so you have a bond involved, right? You have some kind of interaction that is happening, and if you try to visualize, for example, if you try to understand that there are some two proteins interacting with each other, go to a atomic level of these protein, and now see there are bonds that are either breaking or there are bonds that are breaking, or there are bonds, some of the bonds are making, and that leads to the changes in the conformation. So the conformational changes that is happening, so there are some movement that is happening which is leading to the interaction of these two protein. Imagine this thing at the atomic level, where simply there are some bond between carbon and hydrogen, or maybe amine group, or maybe with the water and other things. So try to understand it at a atomic level, and that is what we need to understand when we talk about simulation. So this is not just something happening at the larger scale where we say, oh, I'm so sorry. Okay, okay, wait a second. What have I done? I'm able to change it. Okay, sorry for this thing. Yeah. So yeah, I was talking about the changes that is happening at the atomic level, at the bond level, at the level where we are observing these changes at the atomic level. So there are some, if I talk about as I mentioned, there are some bonding or non-bonding interaction, and we have to understand all these things, and to understand all these things, to understand that because if you visualize a protein is not like that every part of a protein is interacting. There is some base, there is some backbone of that particular protein that make that protein stand, or that hold the integrity, or that hold the structure of the protein. And then there are some parts which are involved in bonding or interacting with other molecules. So every aspect of this protein is important to understand how this particular system is working, or how this particular protein is interacting with other protein, and that is done when we change our PDB file to a topology file. So topology file is basically give you three aspects. That is, one is bond that we talk about at a linear stage or at a distance stage. Then we talked about angles where we have three atoms connected, and then we have dihedrals where we have four atoms connected. So topology file provide you these details. detail. And if I talk about the parameter file, where the that quantify the variables which are used in the force, speed, potential, energy, it gives you parameters such as stiffness, equilibrium value of an angle between three atoms. So basically, there is one thing that is giving you the basic, the backbone, or the actual aspect of the structure, and the other thing is giving you the way it is working, the way it is maintaining, it's holding its structure. I mean, if you try to understand this thing, there are some atomic level aspects, and there are some integrity level aspect. I think I'm not able to give it a good terms, but I hope I'm able to make you understand that one is giving you the the atomic aspect of the entire structure, where we are talking about bonds, angle, and dihedral angles, and one is giving you the parameter that is leading to the equilibrium, or that is maintaining the structure or holding the structure so that some activity can be performed. So this is something is there. Then we have a solvation model. Solvation model is basically providing you the exact environment like the protein is present. So we have explicit solvation model, then we have implicit solvation model. If we talk about explicit solvent model, we have some example like SPS, and these are the most used model, where we have uh, I think sir might have given you some structures on this, and these are basically they are providing you these salvation models are actually giving you the exact scenario how these molecules are present. So basically, they are giving you an environment that a particular protein or a molecule have that leads to its activity. So whenever a protein is interacting with other protein, it is happening in an environment. It is happening in a form of something that make this particular protein act. So for example, if you have a buffering situation, or you have some solvent in which that particular protein. So if you imagine a cell, there are so many proteins, and that is they are performing their activities. So there is an environment which is favorable, leading to the activity of that particular protein. That is what solvation is important. That is what solvation is provided. So whenever we are simulating a system, we need to have exact system of, for example, if I'm talking about protein, and if a protein is present in cell, I have to mimic that system, and solvation model give you that. So that's why we need to know, because most of our biological processes happening in an aqueous condition. These proteins are dissolved. They are not present there in an aggregated form. They are in aqueous solution. So that is what is giving you the solvation effect play a crucial role to determine the molecular conformation, the structural changes, the electronic property, the binding energy, and whatnot. So that is what we do. And if I talk about the explicit solvent model, this is the model that rely on using 100 or 1000 of the discrete solvent molecule. So this model, because this is giving you lot many molecule which is actually present in the real scenario. So if I talk about the real cell condition, where millions of proteins are there, lot of solvent molecules are present. So it is giving you an environment where you have 100 or 1000 of discrete solvent molecule present around your actual structure. So this is definitely used widely because for carrying or simulation solvent, because it is actually giving you a real scenario, and such calculation coverage only slowly, because this is actually making you understand the changes at a very slow level because of the large number of particles involved. So every change is happening at a very slow level, which actually giving you a real scenario, because in reality as well, you have so many molecules that are present, so many other proteins that are present, uh, and a particular protein perform its function. And if I talk about uh, implicit solvent model. So in case of implicit solvent model, there is a continuous medium, having the average property of the real solvent. So we are providing a continuous system. We don't have thousands of molecules present around, and that is trying to understand the real or exact condition of a particular protein that is there in a real system. So it is relatively less computational demanding. It is easy to make because you are not providing thousand or hundred of molecules here. You are just providing a continuous medium where you have a continuous solvent present in the surrounding. So that is what is mostly this is the difference between the two systems. And now if I'm come to the exact, so there are different you have so many softwares or tools that help you perform the molecular dynamic simulation, I talk about charms. But today I'll be taking you through the various molecular simulation step of GROMACS. So you have your prot PDB file, which is a clean file that you have achieved from the, you know, uh, that you have achieved from your. Okay, so this is basically once you clean your PDB file and then you achieve, then you use this prod PDB file to study the simulation. So you have to actually, oh, again it's happening. Okay. So now, when you have your structure, for example, if you look at this side, you have your whole PDB structure, which is free of hydrogen bond, which is free of water molecule. Now, because once you want to remove this thing, but now you have to understand the exact scenario. So this particular stage where you have to add these hydrogen bond, you have to add the solvent, you have to add the forces that are present in the actual real system. So once you are cleaning your entire file and you are making it look like the exact structure, only the structure of that particular protein. But now, because you want to simulate this system, now you will add the hydrogen, now you will add the solvent or the force field that is there in the actual scenario, and that is done through the PDB2 GMX command that help you change the PDB, which help in changing the PDB file format to the GROMACS competent format, and this give you basically a topological. Now you will have a topological file with you. So this prot process grow is actually a topological file of you, which have an extension of topol.top. So you'll understand all these files when you actually use these tools. You'll get to know all these things. So don't think like that whatever we are studying here is something which you have never heard. Definitely, if you have not used these tools, these softwares, you will not be able to relate to it. But once you're going to perform it, you will understand that. Then you do editing. Then you edit your this thing, and then once you add the hydrogen bond, now you want to put a, what you can say. I mean, you have to define the boundary of your system. So now you're going to add a box surrounding your structure, and that is done with and then you'll get a file called prot new box grow. This will give you editing the conformation of the protein in such a way that it is in the center of the solvent. So now your protein is exactly at the center of your solvent environment, and you have provided boundaries to it. Then moving ahead, you have your when you have provided a boundary and your protein is exactly at the center of the solvent, then you will choose a solvation model or a solvent box to create your solvent, to show your solvent. So first you place your protein in the center, and then you give the instruction for the solvent. So for example, if you have water as a solvent, you can add water. Now if you could see, the structure contain water molecule and your protein is inside that. Fine. So this complies basically, this So now you have provided a solvent, then you add ions to it. So you might have some sodium ions present in the system or potassium ion present in the system. Now you try to make your system like a buffer. You try to provide the buffering condition to your protein, which is the actual, which is when you talk about your real cell or your real system, these proteins are present. These protein have their buffering environment. So depending upon your environment, you provide ions to this particular, you know, protein. Now first you have given the first you have given it a solvent, for example water, and now you're adding ions to it. And once you have added the ions, once you have given all the things that is there, that is mimicking the actual buffering system or buffering situation, now you are ready to simulate. Now you have your final structure where you have water molecules, you have your ions, you have other other maybe sodium, potassium, all kind of ions that are present in your actual system. And now you have successfully generated a system for your simulation. So this is basically, first we have actually understand, we try to understand our realistic system, and then now we have simulated. Now our system has been generated, and now we can do simulations here. So now we have the actual scenario generated for us. So there is this term called periodic boundary condition, and why I want to talk, because I have already talked about that we are providing a boundary to a system. We need to locate what is the boundary of our system, and that is what is very important. And why we are talking about the boundary, why we need boundaries to, because if we're not going to put a boundary to our system, we will not be able to understand what all changes are important, what all external forces are important, where is the limitation of our system. That I mean, imagine if I'm talking, if I have to understand a process, we need to understand a realistic scenario. And to understand a realistic scenario, we have some. I mean, if imagine a cell is a I mean it has its own boundary, it has its own limitation. So it has organelles where some function is being performed. It has a nucleus where things are happening in a different way. It has cytoplasm, it has actin, myosin, so many things that are happening. But every everything is happening in a very organized manner. And how that thing is happening, because everything has its own boundary where each protein is performing its function, and not just everything is not happening in a haphazard manner. Everything is so programmed in a cell that nothing is, I mean, imagine how cell is functioning. It is like a very programmed functioning. And why it's happening? Because there is some boundary. There is something that is giving you, okay, fine, this protein has to perform this function. And once the function is performed, this is getting degraded. And whenever there is a need to perform this function again this protein is being made, and it's performing its function and getting degraded. So everything is very systematic, and to have something systematic, you need to have boundaries. So for example, there is a cartoon which is given here. There are some water molecules or hydrogen bond that are essential for the activity of this particular protein. But what if there are some water molecules which are outside the system, which are not at all helping in any kind of bonding, any kind of interaction, nothing. So we need to remove those kind of a water molecule or those kind of hydrogen bond. So once we remove these water molecule, now we have a system, and now we have a system that can actually mimic the exact scenario of a protein. And to have this system, this scenario, I mean, to have, I mean, to understand this but actual scenario, we need to set boundary. We need to remove all the extra water molecules that are present in the structure. So that's why it is very important to have a boundary. And if I talk about the periodic boundary condition are the set of boundary condition which are often chosen for approximating a large system by a unit using a small part called unit cell. So if you could see, this is my unit cell. And when I'm talking about simulation, I will use this as the smallest part and try to build my entire structure, right? So if whenever we are trying to understand a large system, we have to divide it into a small part, and that's how we put a boundary, a periodic boundary condition, so that we can understand a system at a unit level. So periodic boundary condition are mostly used whenever we are performing computer simulation or mathematical modeling. And when I talk about molecular dynamic, simulation, uh, they are usually applied to calculate the property of bulk liquids, crystals, or mixtures. And that is very important to understand to simulate the solvent macromolecules, especially when we're talking about there's so many protein present in the system, but we have to understand a function of one protein only. And to understand the function of that particular protein, we need to have a boundary set for that protein. And that is very important whenever we talk about simulations. So, coming to, I mean, if you could see a concept of central simulation box, and this is the concept of central simulation box. This is actually showing a, a you know, a system at a center, and it has similar systems that are present in their surrounding, which is represented by the light green balls. So the light orange balls, so if you talk about this particular molecule, this is surrounded by orange balls, and it also have green balls that are at its external environment. So we need to study this much only. So we have to understand the interaction of this particular atom with the orange balls only. We don't want to understand its activity with the green ball. So that is something which is giving you, we are actually trying to put a central simulation box. We are trying to put a subject that we need to study or understand in a center, and we are trying to exclude all the external aspect of that particular system, because until and unless we don't understand the smallest unit of that particular system, we will not be able to relate to the larger aspect of it. So that is a concept of central simulation box. We put a box to understand. We put a in we try to put a finite aspect to this infinite dimensions. So, so that is what this periodic boundary condition are providing you. And if I talk about the phase space is another aspect of understanding or putting the, I mean, mimicking a particular system. With the help of phase space, it is a space in which the possible state of a system are represented, with each possible state corresponding to one unit point in the phase space, one unique point. So it can be positional so basically it is providing you the 3D aspect of a system. So you will have, either you have a positional variable where you're talking about something that is present in terms of position, or something that is present in the terms of movement. So there is a position variable and a momentum variable. So at a given atom, if you look at the 3D dimension of this thing, I am right now giving you a 3D dimension, and there can be six, understand this thing happening in a way where you have all the dimension applied. This is not something that is happening. We understand everything in a 3D aspect, but there are other dimensions as well. So if I have to understand the entire system, there are so many things that are there, but right now we are just focusing on the 3D aspect. And when we talk about the 3D aspect, we have some positional aspect where we are talking about the position of that particular protein, that particular atom or a molecule, and there is some changes that is happening due to the movement of that particular molecule, and that gives you a momentum variable. And you have to understand all these things, and once you understand this thing of a given particular, for example, a given atom is represented in a time dependent position or a momentum vector. So you put it like time dependent in case of positioning, that it is present here, and after some time that is present there. So that leads to positional changes. But when we are talking about the momentum vectors, where you have movement as well as, I mean, you have change of I mean distance as well as acceleration involved, then you have your momentum aspects as well. So try to understand the movement happening at the 3D level, not just at the 2D level or something that is happening from one place to another in terms of distance only. So all these kind of aspects are applied with the help of phase space to understand the six n-dimensional plotting domain. So basically, it's giving you a position and momentum of all the atoms that are present. Right now, we are talking about one atom, that there is some position involved, there is some momentum involved. And now try to understand that there are some n atom present in that particular system, and you have to talk about all the aspect of all, you have to consider all the atom and their respective position and momentum aspects. So that is what gives you a phase space, and that is also very important when you are simulating any particular uh, system. So if I talk about the concept of ensemble, that is also giving you a kind of, that is actually, this will help you understand what exactly the phase space means. So we have this in vivo aspect that we are not studying, because there we have a heterogeneous, environment, and we are not studying that. Then we have our in vitro, where we have homogeneous system, where we have homogeneous protein present, and the number of protein is large. But in an in silico environment, we have only single molecule, and then we try to understand the coordinate of this particular molecule. And once we have the coordinate of this particular molecule, we now know how this particular atom behaves, and then we make an ensembles, and we try to put it like a microscopic system. And the property of a microscopic system is basically the average property in all the micro. So we have a microscopic system. When we are talking about a single molecule, it is basically single. So it is giving you a microscopic aspect of that particular system. And when we build this system at a larger scale, we will add these microscopic scale and make it a macroscopic system. So this phase space is actually explaining the macroscopic aspect of a system, because we are not just studying activity of a single molecule. We are trying to understand the activity of single molecule in the presence of n number of molecules. Maybe a similar type of molecule, or even in case of actually representing the real system, it can be heterogeneous mixture as well. It can be different molecule as well. So that is what is something a phase space or ensemble is trying to make you understand. Is imagine the system as a 3D aspect, where so many molecules, even of a same protein, is present, and they are having an effect on a particular single unit of this particular system. So microscopic system is what we build initially, and when we join this microscopic system, when these many microscopic system are joined together, they give you a microscopic view of that system. And that is what we require to understand when we are doing a simulation of a particular system. So ensemble is a collection of all the possible systems which have different microscopic state, but have an identical macroscopic or thermodynamic state. So the if I talk about the macroscopic scale, it is the identical macroscopic scale, but you have different microscopic changes that is happening in this particular in any of the system. So if I talk about different type of ensembles, we have micro canonical ensemble, where we have fixed number of atom, fixed volume, and fixed energy. So what we are doing, we are trying to actually achieve a system with some fixed parameters, and try to look at it as a unit cell, and then we are joining these unit cell to make our 3D crystal. जैसे जैसे अगर मैं एक यूनिट सेल को ज्वाइन करके एक क्रिस्टल बनाती हूँ, ऐसे ही मैं एक यूनिट ऑफ माय सिस्टम को ज्वाइन करके अपना पूरा सिस्टम रिप्रेजेंट कर रही हूँ. So you have different parameters which you want to involve in a particular system. You cannot have all the parameter applied at the same time. You try to mimic this system with a limited parameters. Okay, so because you are trying to simulate it, you are not going to put all the forces, all the possibilities of parameter that are present in a real scenario. You're not going to do it. You're going to put it in a condition where I mean, whatever your requirement is, you try to mimic a system with that particular limited parameters only. So you have your ensembles in a different way. For example, you try to fix the number of atoms. You are putting a fixed volume and a fixed energy. In one case you are trying to fix the number of atom with the volume and temperature, and there is some another case where you have pressure as an important aspect. So you will build your entire system with the fixed number of atom, pressure and temperature condition. And then there may be a system where you have fixed volume, temperature and chemical potential. So this ensemble is actually trying to help you understand the smallest unit of that particular system, and then you are adding this smallest unit and making it at a macroscopic level. So when I'm saying fixed number of atom, that means you have one as a unit system, one atom in your one unit system, and when you're joining all these fixed number of atom, you're making it at a macroscopic scale. I hope you are able to understand. I mean, try to visualize it as something. Uh, I mean, when you are studying, for example, if you want to study a function of a particular protein, you are studying a single protein with the help of in silico approach, but that is not what you need to study. You need to understand what happened when n number of molecule of this particular protein is present in a system. So then you have one unit as a one molecule of this protein, and the entire macroscopic system have n number of molecule of that particular protein, and that is what ensemble or phase space is trying to explain you. So then there is one very important aspect when you talk about molecular simulation is energy minimization, and that is what is very important when we talk about anything that is done in computational aspect. So in the field of computational simulation, energy minimization or geometric optimization is the process of finding an arrangement in the space of a collection of atoms where, according to some computational model, the net interatomic force or each atom is acceptably close to zero. So you have to come to a minimum level of, a minimum level of uh, you have to take your system to a level where you have a force, interatomic force, that is minimum among the atoms, among each other. And the position on the potential energy surface is at a stationary point. So if you could look here, I have given you something. This is basically a local energy minima. This is where we have a local energy. This is what we have a transition point. And whenever you are doing your drug discoveries and something that is giving you a docking, especially in your docking study, this transition point is very important. And then you have your global energy minima. So you try your system to come to a as low as possible. This is basically the minimum level that you can achieve, but you try to put your structure, you try to put your system you try to take your system to a level which is very low in terms of energies. It has to be at the lowest energy, and that is lowest energy possible. That is what energy minimization is. You try to take your system to a level where interatomic forces are least or next to zero, and that is what is your that is mostly achieved when you have a stationary point. That is something where there is no uh, intermolecular forces happening between the two atoms. The collection of atom might be a single molecule or an ion, a condensed phase, or a बायोलॉजिकल मैक्रोमोलिक्यूल भी हो सकते हैं, ट्रांजिशन स्टेज भी हो सकती हैं, कुछ भी हो सकता है. सो बेसिकली यहाँ पे एटम्स कुछ भी हो सकते हैं, सिंगल मॉलिक्यूल्स भी हो सकते हैं, आयंस भी हो सकते हैं. यू जस्ट ट्राई टू पुट you have to put it because that is what is acceptable form of our system. where the energy is minimized. That is what is the natural form, that is what is the native form. जहाँ पर भी एनर्जी की ज़रूरत पड़े, जहाँ पर भी ज़्यादा एनर्जी हो, दैट नॉट गोइंग टू बी योर रियलिस्टिक स्टेट ऑफ योर एनी ऑफ योर सिस्टम. हमारा सिस्टम, अगर इफ आई टॉक अबाउट द रियलिस्टिक सिस्टम, these system are there with the minimum energy, minimum I mean, इमेजिन अगर बहुत ज़्यादा हाई स्टेट ऑफ एनर्जी होगी तो वो सिस्टम एग्जिस्ट ही नहीं कर पाएगा, वो स्कैटर हो जाएगा, या वो बहुत ही इट विल नॉट बी अ स्टेबल सिस्टम. So a system, if you want to achieve a system or if you want to mimic a system, you have to minimize the energy. That will give you the exact or very near, simulation of your real system, because that is how our system exists in our universe or inside our body. फाइन. सो यहाँ पर जो कलेक्शन ऑफ एटम्स की बात हो रही है, कोई भी एटम्स हो सकते हैं, ये प्रोटीन्स हो सकते हैं, ये हमारे मॉलिक्यूल्स हो सकते हैं, कुछ भी हो सकता है इसमें, because we are talking about molecular dynamic simulation, and ये molecular dynamic simulation is not only limited to macro biomolecules or bio macromolecules only. They can be anything. So the computational model of chemical bonding might, for example, be quantum mechanics. Okay, so it can go to it actually generally follow a principle of Newtonian physics, and you actually, especially when we talk about protein movement, we actually follow Newtonian movement only, and we have we our system follow this particular Newton's law of motions only. So we try to, whenever we do computational modeling of any of the protein, we try to use Newtonian physics, and with the help of Newtonian physics, we try to provide a system, we try to take our system to a level where we have minimum energy involved, and that is how we achieve near to the real system scenario. So assumptions are the protein native structure correspond to the state with the lowest free energy and the protein of the protein solvent system. That is what I mentioned, that mostly our system are at the low free energy state. So they are at an energy state which require minimum. They are at the minimum energy state, and I'm not from minimum energy state. I'm not talking about achieving exactly global energy minima. No, it can be here, it can be here, but it cannot be here. If you have a system that is generated here, that will not make a better system. You don't have a better system at that point. You cannot use this system to mimic the exact real scenario. You have to do the in energy minimization so that you can take your protein to a level which is exactly mimicking the real scenario. So energy minimization, we have, as I talk about, there are transition state, there is equilibrium structure, there is equilibrium structure at the global level as well as at the local level. So that is what I mentioned here, that you have uh, a local energy minima, and then you have a global energy minimum. So you will not have the same, you will have equilibrium at the local level as achieved at the local as well as at the global level, as an entire molecule or as a particular atomic aspect as well, at a particular atomic level as well, which means local, which depict local aspect of equilibrium state. So this is something more of the mathematical aspect. I'm not taking into detail because we have limited time, and there are different type of energy minimization. First order derivative, where you have steeper descent, and then there you also have conjugate gradient. I'm not taking this into detail, but I have seen sir's video and I have seen his PPT. He has explained everything in a way that you can able to understand what exactly these things are. And I'm not taking you through because I have limited time. I'm left with half an hour only, and I have to take you through the entire assignment as well. So if I give you a summary of what we have done so far, is that first we are trying to build to simulate any system. We need to build first we need to understand the realistic system, and to understand the realistic system we have to convert our actual pdb file to a clean PDF file, and once we have the clean PDF file, we try to add the hydrogen and try to reach the actual state of this particular protein in a real system. So we add hydrogen, we add our solvent, we add a new box, we define our boundary, we put our protein in the center, we add our solvent, for example water, we add our ion, and now we are ready to go. Now we have given a buffering condition to our protein, and now from here we have to take this system to the lower energy level. So this is the initial coordinate. This is the initial coordinate which is prepared for simulation. It could possibly have a bad contact, causing it at a higher energy and forces. Now we have to perform energy minimization, and that through energy minimization we try to take a system to a lower energy level, and we try to find, first we have to take it to a nearby local minima, and then with the help of with the help of removing other external molecules or the barriers, we have to take it to a state where it has a equilibration aspect, where so equilibration help in escaping the local energy barriers. So we have to break these energy barriers, we have to gradually move it to a lower level. We're not going to take our system directly to the lower aspect. We have to, with the help of, definitely there are some tools that are helping us to do these minimization, and we have different methods where we are using either first order derivative or second order derivative, depending upon the situation, depending upon our experiment. We just need to take this system from the higher energy level to a lower energy level, and it's not going to be a direct system. It will take several steps to achieve the lowest energy state. So further we have now to achieve the minimization. We have, these are the steps that are involved in the energy minimization, especially in GROMACS. So you have your protein solvent ion file, which is your this structure. This is your final structure of a protein which is ready to simulate, which is a final structure, and you give a command of energy minimization, and it will lead to a TPR extension. PR extension contain trajectory files. It give you coordinate, velocity, and forces and energies. So you have then energy file, which is represented by EDR format. It actually take your file to a lower energy level. So whenever you run your minimization, for example, if you are using steepest gradient method, so you run the minimization step, and then you achieve a EDR file, which is your energy file, EM log file that give you details about several energy terms and performance analysis at the end. And TRR extension is something that give you the trajectory, the coordinate, the velocity, the forces, and the energy. All these three files you'll achieve at the end of when you run your when you run minimization step. And these are the following step, and you have different methods involved. For example, as I think we have talked about, especially when we talked about the ensemble, there we have something called velocity based aspect. Then we have NVT log file is same as that of which is giving you energy level. These are different parameters that you're going to put. I am not taking this into details, but still I'm going to tell you that these are the files that give you molecular topology and simulation parameters, and with the help of all the simulation parameters, because first you have got the topological and simulation parameter with the help of these TPR files, and now you want to run energy minimization, and then you get a different, for example, you have different parameters like pressure, temperature, volume, temperature, time dependent temperature changes, time dependent pressure changes, and that is what you're going to change to reach to a minimum energy level. So because these are the forces that leads to higher that took your system to a higher level, you have to change all these parameters so that your system can come to a minimum energy level. And these are done through the different commands. You'll use the software and you'll do this thing, and you have to achieve these files. You have to get these files so as to have your minimized system ready for you. And energy minimization, if you try to look the visualization aspect of energy minimization, that there is this figure give you a normal protein alignment, and this is how the another structure look like. And when you get into detail of this structure, there are some changes required here, because this structure appear to have high energy, and you will make some changes. And this is just a one residue of this structure. This is not the entire protein. So you might need to minimize energy at a particular site of that of a particular, at a particular residue or at a particular domain. You might not have to do everything on the entire structure. So for example, in this case, you don't need to have any minimization because everything is at place. Because can you see there is no defined structure in this particular structure? Because at this stage there are very high energy stage that they cannot achieve a exact structure. But then you have to make some changes. You have to minimize the energy and you will get the exact structure. But in this case, in this particular residue, you don't have to make any changes because everything seems to be at place. Everything is well defined. Every bond, every conformation is clearly visible in this particular state. So you don't have to make changes here, but definitely in these places you need to change because there is some energy minimization required. And that's how the graph look like, where you have to take your reduce your energy to a lower level. This is in terms of time aspect. This is giving you energy minimization graph of time with the help of time aspect. And if I talk about the analysis part of your molecular simulation, where you try to understand whether you have achieved the real state or not, and you try to analyze various various aspect, various parameter that are showing that your model is exact or nearby copy of your real system. So you have this VMD software which help you analyze your molecular dynamics. With the help of VMD, you visualize your molecular dynamics structures, and it also provide a variety of ease to use GUI tools. So you have some, you have your especially when you talk about GROMAX trajectory, you use VMD, and it will help you give study different parameters, especially if you talk about so you need to analyze certain parameter. For example, whenever you are simulating anything, you have some deviations in your system. So you need to study the root mean square deviation. You need to understand, you have to calculate the root mean square deviation. You need to understand whether the exact conformation of a particular residue or a particular amino acid has achieved or not. This you will go into do, you have to put certain mathematical formulas to achieve that, and this is done through you have your softwares attached, that with the help of that you can do this thing. You also do energy calculation, you also do protein ligand distance study, you also do dihedral angle study. And that's how this is what analysis part actually tell you, because you need to mimic your system exactly like a real situation. So you need to understand whether the bonds angles are on the place or not, whether the dihedral angles are exactly all the aspect of, you need to understand the dihedral, you need to figure out the values of dihedral angle of that particular structure. So all these things you're going to do in the analytical part. And with the analysis, you have different analysis. For example, if you want to understand the hydrogen bonds associated with that particular structure, so you will do the hydrogen bond analysis, and for that you have to give different command to get your 2D plot. Fine. So if you have to understand the root mean square fluctuations, you have certain commands. So basically, root mean square fluctuation give you calculate the fluctuation of your alpha carbon in a residue of a protein in comparison with the respective average structure throughout the simulation. So that is also given by a particular mathematical formula, and that is what you're going to give a command to that, and you try to understand what is the root mean square fluctuation of your system. These are some mathematical term that comes in the analysis part, and once you're going to do your molecular simulation, you'll get you will understand all these terms and techniques, and they are different so you just need to have all the thing in place. For example, if I talk about the dihedral angle analysis of a protein, there you have to actually understand a specific residues dihedral angle that you have achieved through molecular simulation, and then you try to, because dihedral angle actually give you the exact conformation of a particular amino acid, and that is what is the basis of your building a whole protein structure. And that is very important to understand the mechanical aspect of a protein, what kind of a movement are involved in that particular protein. So this is what is very important to understand. So you have in analysis part, you have different type of what I say, different aspects that you need to understand, different mathematical calculation that you have to do to understand whether you have achieved the real scenario, whether your model is near to your realistic model. And there are many things that are involved, and for everything you have some sort of commands that you need to give to have your respective parameter aligned, or to have your actual values. For example, in case of dihedral angles, you need to understand what all dihedral angles for a particular residue is there in your molecular simulated structure. So these are the things that you're going to understand once you're going to do these things. And if I talk about the application of molecular simulation dynamics, so definitely it helps you understand the conformation of a protein. And I'm just talking about molecular simulation in terms of biology. So for example, when you visualize a structure, you have your structure backbone. As I also mentioned, there is some aspect of a protein that is holding the structure, and that gives you a backbone of your structure. And there are some residues that is helping in the movement, or which are moving constantly. They must be involved in the interaction with other proteins or molecules, or other biomolecules. So it's not that every part of a protein is involved in a function of a particular protein, or every residue of a protein is involved in making some conformation changes. So you have some backbone which is rigid, which is fixed, which holds the structure of your protein, and then you have some conformational changes that happen due to some residue that are constantly moving. And these residues are mainly involved in the interaction of any of the protein. And with the help of molecular simulation, or with the help of visualization tools, you try to understand these residues only. So as I have shown in the cartoon, you can now actually understand how a particular protein get folded, and that is how your system, that is what you are simulating through the molecular folding of a protein, is what you are simulating through a molecular dynamic simulation. So to understand the folding, you need to understand every parameter. You need to understand a single residue, how it's involved, how many other amino acid it's interacting. And to have all these things, you have to have your topological and parametric file intact, and then only you can generate your system. You need to provide the exact solvent, exact buffering condition to all your molecules. And once you have your simulated system ready, then you change the parameters, like different parameters, and then to try to understand the changes that is happening due to change of a particular parameter. So that is what simulation gives you. That's how you get to know the structure. That's how you get to know whenever you have a mutation in a protein, what kind of conformation changes happen. If you change that uh, a particular amino acid in a protein. So quickly moving ahead with an, oh God, what's wrong with this tab? It's not moving. It's wrong. I think there's some problem with my pen of my tab. Just give me a second. It's not taking to another slide. It's just taking to. Although I have removed the typing mode as well. Wait a minute. I'm so sorry for this. So quickly moving to the practice question, and I'm not going to talk about the practice question in detail because I think it is giving, I have already given all the answer during our discussion. So molecular dynamics simulation are based on quantum mechanics and Newton equation or both A and B and none of these. Can any of you answer this question? Like we talked about this. So it is because of Newton's equation only. It follows the Newton's mechanics, and especially when we talk about protein aspects of computational model, which are talking about the protein simulations, they are followed as a Newton equation only. They follow Newton's law of motion. So when we convert our PDB file to GRO
Uh,
file, which
Shikha,
yeah,
shouldn't it be like both the quantum? Can you just go back to the previous slide? Shouldn't it be the quantum mechanical equation and both, like both A and B Newton's
no, no,
equation?
no, 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 no. We haven't achieved our molecular dynamics simulation with the help of quantum mechanics so far. We haven't reached that level abhi tak. It's just following the Newton equation.
Uh, for some molecular dynamics simulations, I guess quantum mechanical equation is used, right? Like either of the two.
Uh, I doubt that, but I think especially, I think maybe for some it might be because whenever we talk about protein aspect, whenever we do the drug, whenever we talk about the interaction study or simulating something that is relating to protein, we always talk about Newton's mechanics.
Okay,
I
okay.
doubt that is there. I need to check if you're saying so, and if you have the evidence, okay, I'll check that. But as far as I am aware,
No,
molecular
I
mechanics and
don't have
quantum
any
mechanics are
evidence like
different. I mean, sorry, I
I
mean, if
don't.
you are aware of this thing, I mean, as far as my knowledge, as per my knowledge, I know this thing that molecular mechanics is running differently and quantum mechanics is different. Through molecular simulation, molecular dynamics, we haven't, I mean, molecular dynamics simulation is different from quantum. I mean, molecular mechanics is different from quantum mechanics so far. This is what I know. Maybe there are some technology, there are some simulation which are based on both, or but mostly it's on Newtonian mechanics only. I mean, I'll search for it if you're saying so, but I doubt I have heard that.
Okay, okay.
Okay, so it's through that only. I think quantum mechanic calculations are far different from what we study in molecular mechanics or Newtonian mechanics. So I don't know, maybe I mean अभी के लिए मैं ये कह सकती हूँ कि I as far my knowledge it's through Newtonian mechanics only. Okay. Yeah. So next is how do you convert your PDB file to GRO file? This is through the XTC, and I mean whenever you convert it, you'll get which file you will not get. Okay, so you'll get a GRO file, you'll get a topology file, you will get a parametric file. You will not get .xtc. These are the things that you're going to understand when you're going to do, when you're going to perform these, when you're going to give these commands. But this is something I have to, I mean, there's nothing that I can explain in this. This is what exactly is the fact. So I'm not taking it into detail. What is res_nr defines in a topol.top file, that is topology file. And I think you know the answer of this, that and as it is as it is showing the, I mean, this is also giving you a hint that RESN represent residues, and that gives you residue number only, because that is what is required when we are talking about the positioning of any particular residue of amino acid in a topology file, because topology file give you the, uh, I mean, your entire placement of the atoms that is present in your system. Then you have phase space is a 3D dimension space, 4D dimension space, 5 dimension space, or 6 dimension space. And we have discussed this thing as well. So it's a 6 dimension space. It gives you all the possible dimension. It is giving you a it is leading to a microscopic scale to a macroscopic scale of your system, and is giving you the entire aspect that shows the placement of your atom in your system. So it is not just talking about a single atom, it is talking about n number of atoms that are present in your system. So which physical property is constant in micro-canonical and canonical ensemble, and it is atom and the volume. If you go and look back my presentation, you'll get to know. I have told you about the different ensemble where you have different parameter fixed. For example, pressure, temperature, volume, atoms, atom numbers. And when we talk about number of atom and volume fixed, it is happening in the microcanonical and canonical ensemble only. What are the conditions for minima, maxima, and saddle stationary points in a potential energy space, respectively? And I think it's not being okay, huh? So this is also a very factual question, and I have given these value in the presentation. Please check this. This is what we study when we are doing energy minimization. We have a minimum stationary point, we have maximum stationary point, and we have saddle, which is a transitional stationary point, and very important when we are doing a drug designing. Next is, which of the following is known as a bonding energy term? So angle bending term is what is given to a bonding energy term. It actually gives you how the angles are being formed. So it is not just two atom involved. When you have three atom involved and there is a bending of a molecule, of the, I mean, there is a bending involved, angle bending, and that give you a terms in the form of angle, not in the terms of distance. Next is, which of the following is not an implicit solvent model? I have given you the list of implicit solvent model, where you have Poisson-Ball solver, generalized bond model, Karpus EEF1 model, but polarizable Wortel model is not your implicit solvent model. Then you have topological file may specific to bonds 2 atom connected, dihedral 3 atom connected, angles 4 atom connected, or angles 5 atom connected. So whenever you talk about the topological file, it will give you bond 2 atom, I mean, it may specify bond 2 atom connected. Dihedral के case में you have 4 atoms, that's why this option is wrong. Angles के case में you have 3 atoms, that's why these two options are also wrong. So it is kind of a sort of a tricky question. That's why only bond, because in case of bond, 2 atoms are connected. In case of dihedral, 4 atoms are connected. In case of angles, you have 3 atoms that are connected. So that is what is there. What is the post-processing tool of an analysis? I mean, whenever you do your analysis, that is the tool that you use, that is given by TRG-Con. So this is something very factual. This assignment is totally factual. You don't have to, whenever you're going to perform molecular simulation using different, maybe GRUMAX or CHARM or whatever tool you're going to use, you will understand what all these file means, and as such they are just factual aspect of molecular simulation. And in the next, I think we're going to study something about molecular docking and drug discoveries and other things. And meanwhile, I would ask you to go back and look all these. If you can work on these tools, that would be great. You'll understand what exactly we do when we talk about molecular simulation, what all things has to be changed, what all things has to be do, what all commands you have to give to convert your file. And the best part, that the most interesting, when I learned these things during my masters, The best part I really like when is to clean the PDB file, and that is something that was very fascinating to me at that particular stage. So something these are the things that you're going to understand whenever you're going to do the detailed study of these tools or these softwares. So I don't know how much you're going to get through this, but sir has given, I think, something in details. You go back and try to figure out what all you can learn from his lectures. I have very limited time. I can't take every aspect of molecular simulation right now, but if you have any question, any doubt, we can discuss that in next class. So that's all from my side. If you have anything to ask, you can ask, or if you have anything, please go and do read about these things so that you try to relate to whatever you have learned in this today's class. So this is something very important whenever you are learning, because even when I was learning all these things, it was very difficult to understand things initially, especially theoretically. But when we perform these functions, when we gave these, when we perform we use the tool, that's how I also get to learn, and that is what is very important to understand. And especially because at the end of the day, you need to visualize your system, and that is what is very important. Uh, Especially when you are trying to, you know, I mean, there are so many things that are important, not just visualization. You have to, this energy minimization step and everything is very important when you talk about drug discovery, or when you're talking about your, you know, docking your small structure or a ligand to a protein. These things are very important, and going to when if you are working in this field, you'll understand these things and you're going to perform these functions, but you need to have your basics intact so that you know why you are doing that particular why you are doing any particular, you know, task when you are doing molecular simulation. So these are the things that you, I'm not someone who can, I mean, talk a lot about molecular simulation, because that is not what I'm doing right now in detail. But I have gone through this path, and I know that it's a very interesting field to do. It is a very interesting field to study. So, I mean, and really fascinating at times. So, just go back and read about these things. Just go back and try to use the software that are involved, and that is what is required to understand the practical aspect of structural biology. So, that's all from my side today. And if you have anything, just let me know. And thank you for joining.
Thank you.
Yeah, thank you.
So, may I leave?
I mean, if you don't have anything to ask, you may. And but just do read about these things if you are really want to understand molecular simulation. Just go back and try to do these things if you have a chance to get it. I mean, if you have a first hand, I mean, if you want to learn it in I mean, how to use the software, how to use the tool, how to do the simulation, use the software, try to understand the tool, and maybe you can, that would be very helpful
I
for you.
have first-hand experience in molecular docking.
Okay.
Although I was trying for Gromax, but I could not, like, finally when I was doing my project, I could not
Hmm.
like complete it. I just only the installation of Gromax needs so many processes. So
Right,
after that,
right.
so running I did not have that much time to like see the dynamics of the complex I got. So in future maybe I will be trying, and obviously this course will help me, like the
Right.
basics I have learned. I'm not bioinformatics from bioinformatics background, but
Okay.
I try to learn just because of my interest. That's it.
Okay. Oh, that's amazing. That's amazing. That's something very, I mean, you know, amazing to hear that someone is trying to understand the biological aspect of informatic computational studies.
So it is
Fascinating.
actually very important. I mean,
Yeah,
you are from
right,
any free field, like you need to understand. Some computational work will be there if you are like our biology background, like if you are doing any kind of
right,
project. Right?
right. Anyways, good night, Paramita. Thank you for joining, and I hope you have some idea of molecular simulation today with this session.
Yes, yes. I will try to learn. It's not like I have learned everything. Like, just have.
Huh, definitely. I
Um,
mean, that's what I'm saying, that especially when you are studying any of the software or any of the tool, you need to do it by yourself.
yes, yes, yes.
That
Otherwise,
is what is very
you.
important, right?
Yes, you only get to learn when you do things. Otherwise, like it's just like like what happens. I just like get to know. Okay, this file generated. This happened. But
Right,
when I'm
right,
doing
right,
actually, I will get to know. Okay,
right.
thank you, Shikhash. Thank you for the talk.
Thank
Okay.
you. Okay, thank you for joining. Yeah,
Good
you
night.
can.
Good night.
Yeah, good night.